Hello, everyone. In this lecture, I'm going to essentially just explain how synapses work. I'm going to focus on mechanisms and pathways that are more or less universal to synapses. So when most people think of synapses, they think of specific neurotransmitters and their signaling pathways. I'm actually going to avoid talking about those kind of specifics since each neurotransmitter has many different receptors. They each have different splicing variants, different subunits. These pathways are occurring in the context of specific cells. For example, there's estimated to be thousands of neuronal cell types when examined at the transcriptional level. So instead of talking about that, I'm going to talk about a couple of different pathways that are universal to synapses. So some of the more universal aspects include vesicular fusion and recycling, synaptic structural stability pathways, and general heterotypic communications between astrocytes and synapses at the so-called tripartite synapse. So I hope you guys enjoy, and if you have any questions or would like to see another lecture in the future, let me know, and thanks for watching. So we start with our synaptic bouton over here, and here comes along an action potential, right? So the action potential is propagated by sodium influx, which depolarizes our cell, and depolarization from roughly -70 resting membrane potential to +30 is our action potential, and it activates inward rectifying voltage gated calcium channels, or VGCCs. So the activation of these voltage gated calcium channels at the synaptic bouton leads to a massive influx of calcium. So these calcium ions will immediately begin binding to calcium-sensitive proteins. One very important calcium binding protein is this synaptotagmin right here in orange in this figure. So synaptotagmin binds calcium using its C2 domain. So calcium binds to the C2 domain, and this allows synaptotagmin to facilitate the activation of this other complex. This is our snare complex. So synaptotagmin activates the snare complex by displacing a protein called complexin. Right here, you can see it's called complexin. Complexin is also binds to calcium. So calcium comes in and it displaces complexin, or at least reduces complexin's affinity for the snare complex, and it also is directly displaced by synaptotagmin. And so removing complexin from this primed snare complex, you can see complexin's kind of holding together the snare complex. Removing complexin requires two things. It requires calcium and calcium-bound synaptotagmin. Once complexin has been ejected, synaptotagmin is able to come in there and mediate activate the snare complex. Specifically, what we see is that synaptotagmin is able to induce the binding of synaptobrevin right here, this little red guy, the V snare, and then it causes synaptobrevin to bind the plasma membranes syntaxin and SNAP25. So bound to the plasma membrane down here are going to be syntaxin and SNAP25. These are the T snares. So V is our synaptobrevin, and it is on the vesicle. V is for vesicle. And T is for target membrane. And our T snares are syntaxin and SNAP25. And when synaptotagmin facilitates this interaction between these two sets of snare proteins, they pull together so strongly that they actually cause the fusion of their respective membranes, and then the subsequent release of neurotransmitters via simple diffusion. And this process occurs about, on average, only 5 vesicles per depolarization, which I thought was actually pretty low. Also worth noting, we know that GABAergic synapses can inhibit action potentials, and they actually do this by opening chloride channels. So we know chloride is a negatively charged anion, and when GABAergic receptors open, when they bind GABA, they are permeable to chloride, and chloride rushes in, and that would effectively inhibit all these voltage-gated calcium channels. And without calcium, we don't see complexin getting ejected, and we don't see the activation of synaptotagmin. So chloride prevents the opening of these voltage-gated calcium channels because the voltage required, the depolarization required, would be inhibited by chloride. And lastly, I wanted to mention a couple of toxins that are commonly used in research. These include botulinum, tetanus, and alpha-latrotoxin. These are potent paralytics that function at the synapse by basically messing with synaptic vesicles in this fusion process. Botulinum specifically cleaves SNAP25, so botulin cleaves SNAP25. It also cleaves synaptobrevin, so it cleaves the V snare synaptobrevin. And latrotoxin is a calcium ionophore, so it's a calcium channel that causes calcium to come in all the time, no matter what, and this just causes massive release of vesicles. So alpha-latrotoxin basically floods the active zone with calcium, and that prevents, or that just uses up all the synaptic vesicles, and they won't function afterwards. And finally, tetanus is another protease that specifically cleaves synaptobrevin. So a lot of paralytics tend to focus on this transmitter release because it will very quickly stop the heart from beating, because this process is occurring in order for our heart to continue beating and to generate muscle contraction. Once our synaptic vesicle has partially fused with the membrane at the active zone, our vesicle needs to be recovered, restocked with neurotransmitter, then returned to the active zone. So the snare proteins that are kind of zippered up like this, these zippered up snare proteins need to now be untangled in order to recycle our vesicle, right? And this is done by the alpha SNAP and NSF proteins. These proteins are of the AAA, so AAA ATPase family, and they use ATP hydrolysis to disaggregate protein complexes, to break up protein complexes. And that is what they're doing in this scenario. They're basically binding to these snare complexes and they're tearing them apart until you get two separate snare proteins. And so if you don't have this NSF or alpha SNAP protein, you just see aggregated snare complexes all along the presynaptic membrane because they can't be pulled apart. So this is a very important process to disaggregate the snare proteins for continued vesicle fusion. After our snare proteins have been untangled by the NSF and alpha SNAP proteins, our vesicle then needs to actually be pulled back into the synapse, and this occurs through endocytosis. That's what it says right here behind this app. So this is endocytosis. So during membrane fusion, while our snare proteins are fusing with the membrane, something interesting is happening in the background. Kinases localized to the active zone. So these kinases localized to the active zone are phosphorylating phosphatidylinositol at the 5' position. So these so-called PI5Ks, or phosphatidylinositol 5 kinase, are generating phosphatidylinositol 4,5 bisphosphate. And that's, again, a membrane lipid, right? Phosphatidylinositol. That's a membrane lipid in the plasma membrane, right? These are the phosphatidylinositol 4,5 bisphosphates. And via some unknown regulatory mechanism, they only do this once the vesicle has partially fused with the membrane. So the presence of PI45P also called PIP2. You've probably heard of PIP2 from the calcium signaling pathway. So this PIP2 is important because it recruits proteins critical for endocytosis. Specifically, it recruits AP2. AP2 is an adapter protein that binds to PI45P. So you see PIP2 in these little. So actually, PIP2 isn't shown in this graphic, but we have clathrin. So clathrin are these little black T's. The clathrin doesn't actually directly bind to the membrane. It binds to AP2, and AP2 binds to PIP2. AP2 also binds to various N-terminal motifs on transmembrane proteins. So certain receptors have on the membrane side, the cytoplasmic side of the protein, they actually have motifs that bind to AP2. Regardless, AP2 then functions as an adapter for clathrin triskelions. A clathrin triskelion is basically it's composed of 3 clathrin heterodimers. It has a light and a heavy, each subunit has a heavy each clathrin protein has a heavy and light subunit. And there's 3 of them per clathrin triskelion. These clathrin triskelions then oligomerize around the vesicle and they form a cage. You see the cage very nicely right here. They form this cage-like structure. So again, the order of events is a PI5K phosphorylates and creates PIP2 during membrane fusion, and this PIP2 recruits AP2, and AP2 recruits this clathrin coat to initiate endocytosis. At the budding neck of a vesicle, so the neck of this vesicle right here, you see this little blue line right here, and this represents the dynamin protein. Dynamin is a GTPase that oligomerizes around the neck of an endocytosing vesicle. And then dynamin uses the energy derived from GTP hydrolysis to kind of wrench together and to squeeze really tight on this neck of the vesicle until it actually pinches off. We see right here it is pinched off. So another important player in this process are these so-called bar domain proteins, BAR. So bar domains are lipid binding proteins that promote membrane curvature. And they are also critical for the process of endocytosis. So a bar domain containing from a protein like amphiphysin or syndapin. Amphiphysin is the most popular one in research in the synapses because it causes a developmental disorder when it's mutated. So amphiphysin will use its bar domain to produce the proper curvature of for the endocytic process. And there's also evidence that bar domains are specifically localized right next to dynamin. So you have bar domain, like you have a bunch of amphiphysin right here or next to dynamin. So dynamin pinches off the vesicle, and bar domains from proteins like amphiphysin promote the correct membrane curvature in order to produce this process. And then finally, we have phosphatases that will recognize these vesicles and return the PIP2 back into PI4P, so that it'll remove this 5' phosphate that was placed by this kinase. Following endocytosis of our vesicles, they are quickly acidified by a V-ATPase. So this is a V-ATPase. These are proton pumps that couple ATP hydrolysis to proton transfer between membranes. So the acidified vesicle, which now has all these protons, will then have a proton gradient that can then be used by various neurotransmitter transporters to move and stockpile their neurotransmitters in the vesicle. VGlut1, for example, uses a proton gradient to import glutamate. And interestingly, neurotransmitter transporters are often used to identify specific types of synapses. For example, if you want to pinpoint GABAergic synapses in, say, an immunohistochemistry experiment, you would use a fluorescent antibody towards VGAT. Right? VGAT is only going to be found in glutamatergic or GABAergic synapses. Or if you're looking for excitatory synapses, you'd use an antibody towards VGLUT, VGLUT1 or VGLUT2 to give specific, if you're looking at specific subtypes of glutamatergic synapses. Anyways, once the vesicles have been loaded with neurotransmitters, continued calcium flux then causes a protein called RAB3 to bind the vesicle. So I'm not really sure how this happens, but it does happen. RAB3 binds to our vesicles, and as you might know, RAB proteins are a class of proteins that function to direct components of the endomembrane system to different areas. Well, RAB3 directs synaptic vesicles back to the active zone. So in the next slide, we're going to talk a little more about RAB3 and the active zone. So a lot going on in this slide, and this is basically what the active zone looks like. So once you have a neurotransmitter loaded vesicle that has been brought to the membrane, you have all these different proteins that are performing some function in that active zone. So right here, you see our RAB3 that is attached to the synaptic vesicle. We also see our calcium-sensitive synaptotagmin right here, bound to our vesicle. We see our T snares also, synaptotaxin, and VAMP would be synaptobrevin, and our other T snare, SNAP25. So one thing that's missing in this image is our complexin protein. Our complexin is in here somewhere, maybe like right here, that is holding our syntaxin and SNAP25 primed and ready to go, but waiting for calcium flux. Note that this vesicle, it has been situated right next to a voltage-gated calcium channel or voltage-dependent calcium channel. And that's not by accident. So this close proximity is actually accomplished by this RIM MUNC13 complex, also as RIM binding protein, but it's mainly the MUNC13 and the RIM complex that are responsible for putting our synaptic vesicles right next to our voltage-gated calcium channel. So remember RAB3. RAB3 is a GTPase, and when it's bound to GTP, RAB3 can interact with its effectors. It's a RAB3 effector includes the RIM MUNC13 complex. So when RAB3 has GTP, it can interact with its RIM and MUNC13 effectors. RIM and MUNC13 then bind to our voltage-gated calcium channel, and this sets up the entire complex. So when you think of the active zone, think of the RIM MUNC13 complex, because these proteins are essentially linking and bridging our voltage-gated calcium channel to RAB3 positive vesicles. And this allows for very quick snare activation following calcium flux. Neurons that are missing this MUNC13 complex are completely unresponsive to calcium transients, telling us that this anchoring complex, this whole complex right here, is extremely important for synaptic vesicle release and chemical synapses in general. Another interesting interaction going on in this image is this RIM bassoon and piccolo complex. So bassoon, piccolo, and RIM. So RIM, again, is nucleating our this whole complex, but bassoon and piccolo are responsible for tethering the primed vesicle to the actin cytoskeleton. So when you think of the actin, like what the actin complex or cytoskeleton is doing, it's linking to synaptic vesicles through bassoon and piccolo. And so although these two proteins aren't required for vesicle release, knockout studies have shown us that, but they do greatly increase the speed at which the vesicles can be reloaded and recycled. So this bassoon-piccolo complex is important for efficient neurotransmitter or synaptic vesicle recycling. This is just another look at our active zone that has all the same players. Again, this RIM MUNC13 complex and RIM binding protein are critical for nucleating all these different proteins at the active zone. And they do this by binding RAB3. RAB3 sets up this entire process. And they ultimately are putting the synaptic vesicle in close proximity to our voltage-dependent calcium channel. We also see RIM interacting indirectly with the basson-piccolo complex, or basson, and this tethers our complex to the actin cytoskeleton. One interaction I didn't mention was this liprin. Liprin A is these liprin proteins, just multiple of them. They are responsible for tethering the synaptic zone proteins directly to the membrane by binding these transmembrane LAR or LAR proteins. So this is another way that synaptic vesicles are kind of held right close to the active zone in the plasma membrane. The active zone is also commonly called the presynaptic density, and for this reason right here. So look at how electron dense the active zone is. This is a transmission electron micrograph. And just look at how densely packed the active zone is with electrons. That's why it's called the presynaptic density. And so this little area is going to be chock full of these RIM MUNC13 complexes, voltage-gated calcium channels, and everything else that we just discussed. So it's really a busy location in the cell, and that's represented in these electron micrographs where we see a very high electron density. Now we're going to transition into discussing the postsynaptic density and how the dendritic spine is maintained. So there's a lot going on here in this picture, but basically the health and the stability of a dendrite comes down to one thing ultimately, and that's actin. Actin is what provides the structural stability of the dendritic spine, and it might seem kind of obvious, but if you don't have actin supporting the cytoskeleton, you don't have a dendritic spine. So when talking about dendritic spine maintenance, it all boils down to whether actin is polymerized in what's called F-actin, like right here, this is F-actin, or if it's depolymerized in what's called G-actin, right there. So let's talk about factors that promote actin polymerization or inhibit actin polymerization. Let's start with the cadherin junction. Specifically, we have N-cadherins. N is for neuronal, and they're very important for tethering the presynaptic membrane directly with the postsynaptic membrane. And they actually increase in number following depolarization. So the intracellular effector subunit of the cadherin complex is going to be this catenine. Catenine, when bound to dimerized cadherins, so cadherin needs to be binding to its adjacent partner. And when that's happening, it opens up a catenine binding domain. And alpha catenine, there's alpha and beta catenine, but alpha catenine, when it's bound to dimerized cadherins, will actually bind and stabilize F-actin filaments right here. So in addition to simply keeping the membranes in close proximity and promoting neurotransmission, cadherins also promote F-actin stability through alpha catenine. Another protein that functions in a pretty similar manner are these Ephrin B and FB complexes. So presynaptic Ephrin B can extend out and and bind to FB2 receptors on the postsynaptic membrane. And this binding interacts or activates FB or FB2, and FB2 is actually a receptor tyrosine kinase. So when FB2 is activated by FRNB, it begins phosphorylating targets that generally promote F-actin formation. Specifically, one very important target is Cal7 or calorin-7, just abbreviated Cal7. When FB2 activates Cal7 via phosphorylation, Cal7 will then function as a guanine exchange factor for RAC1. So it's a GEF. A GEF means that Cal7 is going to switch RAC1's GDP for GTP. And when RAC1 has GTP right here, it can then interact with its effectors. And RAC1 is the mother of all actin remodeling dynamics. And it basically, if RAC1 is on, it is going to be promoting spine maturation and through F-actin stabilization. So when you think of RAC1 is basically synonymous with dendritic spine stability. When you think of RAC1, think of a successful, stable dendritic spine. GTP-bound RAC1 does this by interacting with two very key and critical effectors. One, RAC1 binds and activates PAC. PAC is a kinase whose activity is dependent on RAC1 binding. And when PAC is active, it phosphorylates and activates LIMK. And LIMK controls actin dynamics by inhibiting coughlin. So LIMK phosphorylates and inhibits coughlin. In coughlin, you might recognize as an actin end-severing protein that cleaves actin or coughlin. This is what coughlin normally does over here. Coughlin is cleaving F-actin and promoting G-actin. That's what coughlin normally does. So inhibiting any of these proteins, any of these proteins, like LIMK or PAC or RAC or even calorin or even FB, inhibiting any of that pathway will relieve the inhibition on coughlin. Indeed, there's many mutations in these proteins that are linked to severe intellectual disability and schizophrenia, and that's because coughlin inhibition is lost. Another protein that is activated by GTP-bound RAC, up here, GTP-bound RAC, is a protein, it's called IRSP53. IRSP53. And IRSP53, when bound to GTP-bound RAC, will then phosphorylate and activate this other complex called ARP/WASP, ARP-WASP. And ARP-WASP is a protein that induces F-actin branching. So actually right here, this will be the WASP complex that's promoting this branching pattern of our F-actin. And that is done by this IRS protein activates WASP, and that produces F-actin branching, and that stabilizes the cytoskeleton and promotes dendritic spine stability. So RAC1 is synonymous with F-actin polymerization through the PAC1 and the IRSP53 pathways. This other protein, RhoA is kind of like its evil twin. RhoA is synonymous with F-actin depolymerization. And RhoA is very similar because it's also a GTPase. So when RhoA is bound to GTP, it can bind and activate this protein called ROCK, R-O-C-K. RhoA bound to GTP can then bind to ROCK and activate it. And ROCK inhibits it should be an inhibition sign. ROCK inhibits LIMK. So if this rho A ROC complex is inhibiting lim-K, it is indirectly promoting covalent activity and actin depolymerization. And although not shown here, ROC also activates actinomycin. Actinomycin is an actin motor protein that induces actin contraction. Right? It's a motor protein that will bind to actin filaments and basically pull them together. And this will cause contraction, and it shrinks the dendritic spines. And that happens due to ROC. ROC can phosphorylate and cause that to happen. So Rho A stimulates dendritic spine shrinkage by binding and activating ROC, which inhibits lim-K, and that allows covalent to start severing F-actin and producing G-actin. Rho A ROC also activates actinomyosin, and this causes contraction of actin. So being a GTPase, that means Rho A activity is thus dependent on GTP for activity. Again, we said this GTPase, so it needs GTP in order to do all this. And there are two important Rho A gaps, guanine activating proteins, that hydrolyze Rho A's GTP into GDP and turn it off. The two proteins that turn off Rho A and thus prevent actin depolymerization are P150 and OPH1, OPHN1. So OPHN1 is very interesting because its mutation actually results in constitutively active Rho A and thus ROC-mediated depolymerization. So OPHN1 is linked to constitutively active Rho A because it can't remove its GTP. And not surprisingly, OPHN1 mutations cause mental retardation, likely because of this specific loss of function interaction. The other Rho A gap, P190, is kind of cool because its activity is basically based on integrin attachment to the extracellular matrix. So when integrin is attached to an extracellular ligand, it activates this kinase called ARG, and ARG phosphorylates and activates our Rho A gap, allowing it to inhibit Rho A. Okay, the final complex in this image is this NMDAR complex. And this is kind of the classic postsynaptic density that we're going to discuss more on the next slide. So NMDAR is, of course, a glutamate receptor embedded in the membrane, and its intracellular domain is linked up in this postsynaptic density that is primarily nucleated by this PSD95. So PSD95 stands for postsynaptic density of 95 kilodaltons, and PSD95 uses numerous PDZ domains to recruit all these other proteins that are also found in the postsynaptic density. The two most important proteins nucleated by the PSD95 protein are the Schenck and the Homer complex. The Schenck and the Homer complex. And these two guys, they do many things, but one of the important functions is that they recruit and they stabilize F-actin. So Schenck-Homer complexes can stabilize F-actin. We know this is important because mutations in Schenck are associated with autism and many other mental disabilities. So Schenck is very important for linking and supporting F-actin dynamics as well. And well, also lastly is this other protein, Drebrin. Drebrin is an F-actin stabilizing protein that will bind to F-actin and make it stable. I don't really know how it fits into the grand signaling scheme, but it is there, and it is important because it's implicated in Alzheimer's disease, where it's about 81% less abundant. And some researchers think the lack of Drebrin expression leads to F-actin instability. Okay, so in the next slide, we're going to talk more about this postsynaptic density. We're going to talk more about that. But in this slide, the main point here is that Cal7 activated RAC1 promotes F-actin and spine stability through PAC, and IRS-P53 leads to the WASP and the F-actin branching. And then F-actin is destabilized by Rho-A, which binds and activates ROCK, and ROCK promotes Coughlin activity right here. So this is a close-up of a typical postsynaptic density from an excitatory synapse. So as you can see from the presence of glutamate receptors, we have NMDA receptors, we have metabotropic glutamate receptors, and we have AMPA receptors. The postsynaptic density is primarily brought together by this protein complex that I mentioned in the last slide, is this PSD95 right here in green. So PSD95 nucleates this large complex through these PDZ domains. And I'm guessing the PDZ domains are these green bulbs are these PDZ domains. And each PSD95 has 3 PDZ domains that can bind to proteins like CAMK2, calmodulin kinase 2, or GCAP. GCAP is important because it basically bridges PSD95 to Shank and Homer. So GCAP will bind to Shank, and this is how Shank is recruited to the postsynaptic density. And CAMK2 you probably recognize as a critical calcium-dependent kinase that promotes long-term potentiation. And so by localizing CAMK2 right below the NMDAR, you have a better chance of it being activated. Another important interaction is that Homer can actually bind to IP3 receptors from the endoplasmic reticulum. So if we're talking about a neuromuscular junction, that's going to be very important because you have the sarcoplasmic reticulum that's going to be anchored right next to the postsynaptic density thanks to Homer. Anyways, I don't exactly know what else to say about this slide other than to know that PSD95 is a critical structural protein that's bound to the intracellular parts of glutamate receptors. And this Shank/Homer complex is also critical for constructing and recruiting other postsynaptic density proteins and a lot of these signaling events that happen downstream, such as calcineurin, protein phosphatase 1, and IP3R receptors. So the Shank-Homer complex is also an incredibly important scaffolding protein. Also worth mentioning are these GABAergic inhibitory synapses. So most of what we've been discussing so far really only applies to excitatory synapses. The big player in inhibitory synapses are these Gefferin proteins. So Gefferin proteins are essentially the PSD95 of inhibitory synapses. That is, Gefferin functions to hold GABA receptors at the postsynaptic densities and then nucleate many of the other proteins of the postsynaptic density in inhibitory synapses. In addition, if an experimenter or a researcher wanted to look for inhibitory synapses via immunohistochemistry, for example, an antibody towards Gefferin is what would be used. And by that same token, if an experimenter wanted to look at excitatory synapses, they would use an antibody towards PSD95. In this figure, you can also see these little colibistrin proteins. Oops. Right here. Colibistin proteins, these little guys are responsible for actually recruiting and bringing Gefferin to the postsynaptic density. And the reason colibistin is worth mentioning is because there's certain mutations that are linked to intellectual disability in these colubistin proteins. Now let's zoom out a bit and let's look at the role of astrocytes in the maintenance of a synapse. So this image is a mesh of two different figures, as you can see, but the idea is the same. So let's begin by picking out some of the important metabolic support roles that synapses are playing. Up here in the top right, we see the astrocyte neuron lactate shuttle, which I do have a lecture on if you're interested in. But the basic idea is that excess pyruvate is converted into lactate through lactate dehydrogenase, and then transferred to neurons by these MCT transporters. And once in the neuron, lactate can be converted back into pyruvate. Right here, it can be made back into pyruvate and then shuttled into the mitochondria for energy. The astrocyte neuron lactate shuttle is generally thought of as a major metabolic support pathway because astrocytes not only have first access to glucose from the bloodstream, but it's also the site of lipid droplet accumulation. So it's not shown in this figure, but this is where astrocytes are, where lipid droplets accumulate, and that's where you can basically making lactate from the acetyl-CoA that comes off of those fatty acids. So a lot of the reduced carbon in the brain is derived from astrocytes, since astrocytes are the sites of lipid storage, and they are the primary sites of cholesterol synthesis as well. Since cholesterol is a downstream of lipid droplets or triacylglycerols. Astrocytes are also directly regulated. They also directly regulate the synaptic cleft. So this is the synaptic cleft right here, and they regulate this this region through the by removing excess glutamate from the synaptic cleft. And they do this through the EAA2 transporters. And once glutamate is inside of the astrocyte, it is converted into glutamine by glutamine synthase. And this is perfect because glutamine can be returned to neurons without activating any glutamate receptors. Right? Glutamine is transferred back to the neuron and converted into glutamate, where it's now, bioactive and can activate receptors. And this is why this pathway is needed, because if glutamate was taken up and then released as glutamate again, it would just activate more glutamate receptors and potentially cause excitotoxicity. See, astrocytes also are responsible for regulating extracellular potassium concentrations through these KIR 4.1, KIR for potassium inward rectifying 4.1 channels. And the importance of these channels is again illustrated by the many multitude of syndromes that can result from KIR 4.1 mutations that are almost all related to intellectual disability, hearing loss, and ataxia. And that is thanks to this KIR 4.1 protein. In fact, a common canine spinocerebellar ataxia disease is derived from mutations in the canine KIR 4.1 gene. So defects in KIR 4.1 calcium buffering are very important. They're also implicated in Huntington's disease. A mouse model of Huntington's disease shows defects in this KIR 4.1 potassium transporter. So it's a pretty fascinating transporter. Astrocytes can also directly signal to synapses using so-called gliotransmitters. These gliotransmitters, here on the left, we see a whole host of them, and they can influence synaptic strength and excitability. Thrombospondin 1 and 2, THBS 1 or 2, can bind to NLG, neural ligands. that span the synaptic cleft and can actually promote their successful binding. So let's see if I can go back to the previous slide. Can I go back to the previous slide? I can show you. So we see right here that neuroligands, right? Oh, up here, sorry. Neuroligands can bind to TSP1, and this can promote their successful signaling to the excitatory between the presynaptic and postsynaptic membrane. Okay, what else do we have here? We also have tumor necrosis factor, TNF alpha, right here. Tumor necrosis factor is really important, and it induces AMPA receptor endocytosis, and it's also been implicated in various neuroinflammatory diseases or pathologies. Another important player, which I don't think is shown here, is ATP. ATP is probably the classic gliotransmitter, astrocyte derived transmitter, and it activates P2Y receptors, and that's really, really important as well. And also another thing not shown here is serine. Serine can be released and is actually required for NMDA receptor activation. And there's evidence that serine is actually basically the limiting factor in NMDAR activation. So glutamate won't signal for calcium influx unless there's serine, and serine is derived from the astrocyte. So that's a pretty important process. So it's still not understood exactly how gliotransmitters are released from astrocytes, but it is likely calcium dependent. So astrocytes show very distinct calcium waves, and these calcium waves are derived from IP3R receptors in their endoplasmic reticulum, and also from TRP channels. And these calcium waves lead to snare-dependent exocytosis of vesicles containing gliotransmitters. So it sounds a lot like vesicle release in neurons, but it is distinctly different. But it does require calcium. It does require snares. But other than that, not much more is known about gliotransmission. So just to summarize, astrocytes do many important things, but some of what we discussed are glutamate recycling back into glutamine and return to the neuron, metabolic support through the lactate shuttle, and cholesterol transfer, can provide cholesterol for membranes. Synaptogenesis through thrombospondin. Thrombospondin is very important for linking up these neuroligands. Extracellular potassium maintenance, ion concentrations as well, and various cell-cell communication events with TNF alpha and L-serine binding glutamate receptors, and I'm sure ATP binding P2Y receptors, and I'm sure there's many other that are yet to be discovered. Anyways, this is my last slide, and I hope you guys enjoyed this lecture. If you have any questions at all, I would love to hear them. I'd love to talk about it, and thanks for watching.